Yes, thank you so much and welcome everyone to our next webinar in monthly series Unravelling T Cell Recognition, Insights from Immunology and AI. My name is Hashem Kui and I'm delighted to be moderating today's session. So this series takes place on the first Monday of each month, where we explore cutting-edge perspectives at the intersection of T-cell immunology, AI, and machine learning. Our goal is really to foster an open and inclusive community for everyone interested in T-cell immunity and bridge the gap between academia and industry. Announcement for this series and speakers are shared in LinkedIn and also a dedicated mailing list that we have created for this. As you can see, I'm also very excited to announce that we will be hosting a very interesting and fascinating conference and a hackathon later in September. So I'm looking forward to welcome you all to this event. With this, today, it's my great pleasure to have Dr. Jose Cabezas Capellaro from Professor Dushak's lab, who will be talking about enhancing T-cell affinity discrimination to reduce the cross-reactivity of T-cell therapies. So just a short bio about Jose. He received his BSc in biological science at the University of Edinburgh before moving to University of Oxford to pursue DPhil in interdisciplinary bioscience. He joined the group of Omar Dushak to study T-cell cross-reactivity, where he continued to work as a postdoctoral researcher. Very welcome, Jose, and I'll stop sharing, and the screen is yours.
Perfect, thank you. Sure. Okay, so hopefully everyone can see my screen and the pointer. So yeah, thanks very much to Hashem for inviting me to give this seminar. I'm gonna be mostly presenting work from my PhD at Omar Dushek's lab in the Dunn School of Pathology, where I still am working as a short-term postdoc to finish the revisions for all this work. And I will say, if you're interested, you can find the work on bioRxiv and hopefully very soon published version as well. I just wanted to say we have a lot of time, so if anyone has any questions, please feel free to just shout them if anything is unclear. Otherwise, we'll also have time at the end for more questions. So the aim of my PhD project has been to investigate the role of T-cell ligand discrimination and cross-reactivity and the implications for safety in T-cell therapies. So I'll just start with some brief background. And actually, it was first hypothesized a long time ago in my department by Don Mason in 1998 that if a single T cell could only recognize a single peptide, a mouse would need a box 70 times its size just to fit all the T cells you would need to recognize every potential immunogenic peptide. So of course, we just have a limited number of T cells that have to recognize a much larger universe of potential pathogenic peptides. So this is why cross-reactivity really is an essential feature of natural immunity to give us this full immune coverage. And since this first sort of mathematical calculations, it has now been shown experimentally by different groups that a single TCR can recognize between 100,000 and 1 million different peptides. So this really challenges the view of one T cell, one specificity that has been used in books and lectures for a long time. Now, as I mentioned, this cross-reactivity is essential in sort of a natural immune setting, but it doesn't mean that it's necessarily good in the therapeutic settings. So there are some therapies called adoptive cell transfer therapies, which are currently being used in the clinic, where you can take a sample of a patient's blood and isolate their T cells. And then you can engineer these patients' T cells with a new receptor that specifically binds to that patient's cancer cells. And then you can expand these engineered T cells in the lab and give them back in the patient to help fight the cancer. But as I said, if a single T cell can recognize up to 1 million peptides, when we're engineering these T cells with new specificities, we don't we don't want them to activate against 1 million things. We only want them to activate against the one cancer target peptide. So here, the cross-reactivities can be dangerous. And in fact, unfortunately, there's been multiple clinical trials over the last decades where patients receive their own cells engineered with new T cell receptors that were meant to be activated against cancer peptides, but these cells went on to become activated in healthy tissues and unfortunately led to multiple patient deaths. So this is why for my whole PhD, I've been focusing on developing methods to reduce cross-reactivity in the context of these adoptive cell transfer therapies. So just to give you a bit more background of what the clinical pipelines have been like and what companies that are developing these therapies are doing, essentially you start with a cancer antigen that you want to target, and you can identify a peptide that is presented by the patient's MHC. Then the next step is to identify a TCR that can bind that peptide in that particular MHC context. And then what you want to do is you screen for TCRs that can bind that peptide but cannot bind other healthy peptides presented on the same MHC. So essentially, it's a bit of a trial and error where you can start with a library of TCRs and you sort of identify one that binds your target peptide but not your off-target peptide. And what I will say here is that this is a very challenging process. And this is something that clinicians and pharmaceutical companies struggle a lot with. Because obviously, if one TCR can bind 1 million peptides, you cannot manually identify the identity of those 1 million peptides. So for these clinical trials that failed, actually very little work was done to assay the cross-reactivities of those TCRs. I think the FDA back in the day only required you to test your T cell receptor against a panel of selected cell lines. So a few cell lines from the heart tissue, a few cell lines from the brain tissue, and so on and so forth. But unfortunately, this does not recapitulate the full sort of immunopeptidome diversity in a whole human body. So it's really, really hard to identify these cross-reactivities until this therapy is given to a patient, and then you can be absolutely sure if it's safe or not. So since we hypothesized that it's too complicated to find a TCR that truly only binds one peptide, and there's always going to be cross-reactivities, we decided to instead develop methods to engineer T cells to be more specific themselves, regardless of the binding specificity of their TCR. So as I mentioned before, one TCR can bind 1 million peptides, but that doesn't mean that the T cells will become activated against those that entire set of 1 million peptides, because T cells themselves have different processes to make decisions of when to activate or not. Another important bit of information is that even though one TCR can bind 1 million peptides, not all binding events are equal. So just by pure sort of protein binding rules, you would expect that there's very few peptides that can bind a TCR with a very strong affinity. So this means high affinity means that the TCR will be bound to that particular peptide for a long time. There's obviously only a few sequences for any particular TCR that will give that perfect complementarity and therefore result in a very high affinity interaction. So instead, we consider this to be a continuum. So as you reduce TCR affinity for peptides, you have a larger number of peptides that can bind with equal affinity. And eventually, there'll be many, many thousands of peptides that bind a TCR with very weak affinities. But as I said, only very few peptides that will truly bind with a very strong affinity. And as I alluded to before, T cells are constantly making decisions about when to activate or not. And in fact, T cells are already pretty good at ignoring the weakest affinity interactions. So that if a TCR only binds a peptide very transiently, T cells are able to filter that signal out and not activate. And this is what we call as a property T cell ligand discrimination. And this means that T cells are able to discriminate between high affinity and low affinity interactions. And it's what we're trying to exploit further to improve the safety of these therapies. So essentially, what T cell ligand discrimination is controlled by is the signaling pathways that start at the TCR and go on to activate the T cell. So when a TCR binds a particular peptide, there's a series of enzymatic steps that need to occur before the T cell can be activated. And it has been shown that if the affinity is too low, then these steps cannot happen while the TCR is engaged. And this is the basis of why T cells can ignore low affinity interactions. So once again, for my PhD, we were trying to look at these signaling pathways and try to identify different nodes that we could modulate to make the discrimination stronger and reduce the cross-reactivity of T cells. And finally, I will also mention that we have a few different genetic tools at our disposal to achieve this. So I'll be mostly talking today about knockout experiments I've done, where obviously you can just genetically modify T cells to abolish expression completely of a particular protein. But if you really wanted to fine-tune signaling, we have also used siRNA knockdowns. So essentially, you can express a small RNA molecule in T cells that is complementary to your gene of interest's mRNA, and this will reduce expression of your gene of interest without completely abolishing it. So it's another way to fine-tune the signaling. And finally, we also overexpress genes. So particular genes of interest in the T cells, we can introduce them with lentiviruses to be expressed de novo or at higher levels than before. Okay, so I'll just give you a brief run through the methods that we use before I jump into the data. So all the experiments that I've performed, we follow sort of the standard clinical pipelines of how T cell products are generated, even though we are a preclinical lab. So we obtain blood samples from the hospital, from healthy anonymous donors, and we can isolate their T cells. And as I mentioned, we can knock out specific genes of interest that we want to study with CRISPR-Cas9. And then we infect these T cells with lentiviral particles that encode our TCRs of interest. And then we can expand these into large populations of genetically modified T cells. And these TCRs that we use, I'll go into more detail soon, they bind specific cancer peptides that have already been identified. Okay, so to measure ligand discrimination, we need two things. So firstly, we need a panel of peptides of defined affinities to a particular TCR. And to do this, we use a technique called surface plasmon resonance. So this is a biophysical technique to measure protein-protein interactions. And essentially, we use a chip where we immobilized particular peptide MHC molecules. And then over this chip, we can flow increasing concentrations of our purified TCR of interest. And by measuring the reflection of light, you can get a measurement of how much protein is bound to your peptide MHC. And then secondly, we can take these peptides that we've measured affinities to and pulse them on cancer cells. And then we can measure the actual T cell responses against different peptides and compare them to the different peptide affinities of this TCR. And to measure T cell activation, we do a variety of techniques, such as measuring the amount of cytokines secreted by the T cells, measuring the target cell killing, or measuring activation markers by flow cytometry on the T cell surface. So as I mentioned, we used surface plasmon resonance to measure the affinities of a panel of peptides to our model TCR. So our model TCR is called the C259 TCR, and is specific to NY-ESO, which is a cancer testis. Well, it's specific to a peptide derived from NY-ESO, which is a cancer testis antigen. And this specific TCR has been used in phase 3 clinical trials by GSK and Adaptimmune for sarcoma patients. So what we did is we measured the affinity to NY-ESO and to a series of single point mutants. And what you can see here is that this TCR binds the wild type peptide with a very strong affinity of about 1 micromolar. And as we introduce different single point mutations, you can gradually reduce affinity up to 150 micromolar for the lowest one we measured here. Okay, so now that we had this model TCR and a panel of peptides with defined affinities, we could investigate how T cells make decisions to activate or not against these different peptides of different affinities. So the first approach we decided to investigate was to modulate the CD8 co-receptor. So the TCR directly binds a peptide in a particular MHC context, and that helps the T cells recognize their targets. But T cells have different co-receptors such as CD8, and CD8 in particular can bind MHC in a peptide-independent manner. And CD8, when it's bound to MHC, recruits the LCK kinase in close proximity to the TCR. And the LCK kinase is responsible for phosphorylating the intracellular tail of the TCR, which initiates signaling. So we decided to knock out the CD8 co-receptor and investigate how this affected T cell activation and ligand discrimination. So what we did, as I explained before, is we obtained T cells from blood donors, engineered them with our TCR of interest, and either maintained the endogenous expression of the CD8 co-receptor or knocked it out by CRISPR-Cas9. And you can see the activation of our wild type and knockout T cells against different peptides summarized in these graphs. So each graph is a different peptide titration. And what you can see is the wild type cells in black and the CD8 knockout cells in red. So here, for the highest affinity peptide, you can see that there is no difference in activation between wild type cells and CD8 knockout cells. However, as we reduce peptide affinity, you can see that CD8 knockout cells become gradually worse than wild type cells. And we can summarize this by taking the fold change in activation between the wild type cells and the knockout cells, which is in this graph here. And we can plot that according to peptide affinity. So once again, for the highest affinity peptide, the fold change is 1 because there is no difference in activation. But as we reduce peptide affinity, CD8 knockout T cells become gradually worse at activating. And for the lowest affinity peptides, they're about 100 times worse than wild type cells. And because the slope of this line is very significantly different than 0, we can conclude that knocking out CD8 improved T cell ligand discrimination because it selectively abolished activation against low affinity peptides only. And then we followed the same approach to test different co-signaling receptors. And what we show in these plots is that not everything that is involved in T cell activation will modulate ligand discrimination. So for example, in this plot here on the left, we knocked out adhesion receptors. So LFA1 and CD2 help T cells adhere to their target cells. And this helps them activate as well. But when you knock out these adhesion receptors, what you can see is that T cells become worse at activating against any peptide, regardless of affinity. It's just they blanketly become about 10 times worse without actually modulating discrimination or cross-reactivity. And then here's the results I just showed you with CD8, where discrimination is enhanced. And I just wanted to highlight that we have also identified some genes that have the opposite effect. So for example, here I have CD5, which is a negative regulator of T cell activation. And what we find when we knock out CD5 is that the activation against the high affinity peptides are not changed. So the fold change is 1. But now when we knock out CD5, the activation against the lowest affinity peptides actually becomes better. So we would say that knocking out CD5 decreases ligand discrimination. Okay, so I've just shown you here a few highlights of the molecules that we've tested. But just to summarize that different co-signaling receptors could modulate activation to all peptide MHCs or selectively to high versus low affinities, as we've shown with CD8 and CD5. Okay, and of course, CD8 was the most exciting result we had because it was a molecule that we could knock out to enhance the discrimination of T cells with this model receptor that we used. But then we just wanted to show whether this was reproducible with different TCRs. So the A3A TCR, once again, also used in clinical trials by Adaptimmune. But unfortunately, as I mentioned in my introduction, even though this TCR was designed to recognize MAGE-A3 in melanoma patients, the first people that received this therapy died due to cross-reactivities in the heart. And this was then identified to be due to activation with a peptide from a protein called titin. We know that this TCR binds MAGE with higher affinity than titin. So we decided to investigate whether knocking out CD8 could abolish this cross-reactivity. So once again, what you can see in this plot here at the bottom is the activation of wild-type cells in black versus CD8 knockout cells in red. And the activation against the high affinity MAGE-A3 target peptide is unchanged when we knock out CD8. But now when it comes to titin, even though wild-type cells can activate very strongly, CD8 knockout cells cannot activate at all. And we know from subsequent studies that it was this activation against titin that caused the patient's deaths. So we hope that maybe using strategies like these could have prevented those lethal cross-reactivities. Okay, so we showed that by knocking out CD8, we could enhance discrimination, and we decided to continue testing whether we could increase this effect further with additional modifications to have even less cross-reactive T cells. So just a little bit of background biology. And this is from Alfred Singer's group from about 10 years ago. What they discovered is that during T cell development, T cells need to know that they are binding peptide MHC molecules. And this is why co-receptors such as CD8, which bind MHC, can provide that information. During development, because TCRs are randomly generated, actually a lot of these TCRs could bind folded proteins like antibodies. But these T cells do not survive thymic selection because they cannot become activated. And they identified that this was because of the co-receptors. And in summary, what they found is that because the co-receptors bind LCK, if the co-receptor cannot co-localize with the TCR, LCK is effectively sequestered and the T cells cannot signal. So to put this in a different way, the co-receptors can sequester LCK if they, if the co-receptor doesn't localize with the TCR, LCK is sequestered. And if the TCR doesn't bind a peptide MHC molecule, the co-receptor cannot localize with the TCR. So essentially, we decided to use this bit of biology as a tool to engineer the discrimination further. So obviously, the TCR that we have been using for our model system binds class 1 peptide MHC. So CD8 can promote signaling. But we decided to engineer cells to express the CD4 co-receptor, which has an analogous role to the CD8 co-receptor because it can bind LCK via its intracellular tail. But because its extracellular domain binds MHC class 2, it cannot co-localize with the TCR. And therefore, we hypothesized that it might sequester LCK and increase discrimination further. So to do this, what we did is we engineered the lentiviruses that we used to express the TCR to also encode the CD4 gene. So now we could generate T cells that express our TCR of interest and CD4. And we could also combine this with the CDN knockout that I already described. Okay, so first I'm showing you here what is the effect of simply expressing the CD4 co-receptor to sequester LCK. So what you can see here is the wild-type cells in black and the CD4 overexpressing cells in red. And once again, there is no difference in activation for high-affinity peptides. But similarly to what we saw with CD8 knockout before, when we introduce CD4, we selectively inhibit activation against the lowest affinity peptides, shown here in this plot. And then finally, we decided to combine both modifications. So knocking out CD8 and introducing CD4 to try to achieve the largest effect. And we term these T cells CD8 to CD4 switch T cells because we switched the CD8 co-receptor for CD4. Okay, and that data is shown here again with the same peptide panel. And once again, there's a very large effect against low affinity peptides. So basically, you can see all the summary data in this plot here with the fold changes. So just to summarize, when we just express CD4, we make T cells about 10 times worse at activating against the lowest affinity peptides. When we knock out CD8, shown here in blue, we make them about 100 times worse at activating against the lowest affinity peptides. And when we combine both modifications, so knocking out CD8 and introducing CD4, we make our T cells up to 10,000 times worse at activating against the lowest affinity peptides. So a very, very large increase in discrimination. Okay, so all the data that I've shown so far was generated with a defined 7-peptide panel. So we decided to test whether we could reproduce the same results with different peptide sequences. So as I mentioned, this C259 TCR, which recognizes NY-ESO, has been used in clinical trials before. So there are different groups that have already performed different studies to investigate what potential peptides in the human body this TCR might cross-react with, because those are the ones that could cause lethal autoimmune cross-reactivities. So here in this table, I've got peptides that were identified by a previous group that are cross-reactive to this TCR. And you can see they have very different sequences to NY-ESO, but they all have the same tryptophan residue at position 5. So that is a common feature of all the peptides that were identified to bind to this TCR. So then we used surface plasmon resonance once again to obtain affinity values of these self-peptides against the C259 TCR, and then we carried the same experiments as before. So basically what we can show is that we obtain the same results, which is an improvement in discrimination when we knock out CD8, and a larger improvement in discrimination when we perform the co-receptor switch. But this is interesting because we know that these are the peptides that could potentially be an issue for this TCR. And once again, we can show that we can make this TCR safer while maintaining very strong killing against the target NY-ESO peptide shown here. Okay, so I've discussed a lot about ligand discrimination and this relationship between affinity and activation. But really, the point here is that we propose that if you increase ligand discrimination, you reduce cross-reactivity, because as I said before, most peptides that a TCR can bind with will be of low affinity. So we view it sort of in this cartoon model as in T cells can bind many peptides, and if they have the CD8 co-receptor, they can activate against many of them. When we knock out CD8, the TCR can still bind to the same number of peptides, but the T cells can only activate against a few of them because LCK is no longer actively recruited to the TCR. And finally, when we introduce CD4, even though the TCR can bind to all these peptides, now the T cells really will only activate against the very, very highest affinity ones, because not only is LCK not actively recruited to the TCR, but also is actively sequestered away from it. So this is the relationship between ligand discrimination and cross-reactivity. And obviously, because I've only used selected panels of peptides, we wanted to show that this actually works in a sort of unbiased manner with peptides of completely random sequences. So to do this, we obtain these peptide libraries that are completely randomly synthesized, but they have a preferred length of peptide per library. So we have an 8-mer library, 9-mer library, 10-mer library, and so on and so forth. And we can use these pooled peptide libraries, pulse them on cancer cells, and look at T cell activation. And what you can see is that actually T cells engineered with this TCR mostly only activate against a 9-mer peptides. So it has been shown before in the literature that some TCRs have a length preference, but you get some activation against a completely random 9-mer peptide mixture. And then when we perform of genetic modifications, we can see that this activation against the random library is reduced when we knock out CD8, and is reduced even more when we perform the co-receptor switch. And this is because, once again, we expect that all these interactions are quite low affinity because it's just a completely random peptide mixture. So it's just another way to assay the cross-reactivity of our engineered cells. And finally, we have one more method of assessing cross-reactivity, which is a positional scanning library. And in collaboration with other people in our group, we established this method to measure TCR peptide MHC affinities in a high throughput manner. So just to put it into context, the NY isopeptide is a 9-mer, and these are shown in blue are the 7 peptide panel that I tested throughout my PhD. So obviously, it's a very small representation of the entire sequence space. So what we decided to do is, sorry, and I should also mention, just measuring these 7 peptide affinities was already a lot of work, sort of a multi-week experiments for all of these peptides, because you have to purify the TCR and manually purify each peptide MHC with a different peptide loaded into them and refold them and run the assay. And the machine, the surface plasmon resonance machine that we use, which is a Biocore, which is the sort of the industry standard, can only it only has 4 flow cells per chip, so you can only measure 4 different peptides at most per run. So it is a very time-consuming process. So instead, we decided to move into sort of a different assay using this Cartera machine that you can spot up to 386 samples per chip, enabling you to read hundreds of peptide MHCs per run just by flowing the TCR over all of them at once. To do this, of course, still the most time-limiting step was to purify these hundreds of peptide MHC molecules. So in the workflow that we designed, we used stabilized MHC molecule, a disulfide stabilized MHC molecule. So essentially, this means that we can purify an empty MHC, and we can just incubate it with a peptide to load it instead of having to manually refold the hundreds of them at a time. So this is what enabled us to do this larger scale measurements. What is shown here is just some quality control of these measurements that either with the wild type MHC or with the disulfide stabilized MHC, you obtain the same affinity values for a particular TCR. Okay, so this is the entire data set of the C259 TCR to every single peptide mutant from the cognate NY isopeptide. So this matrix shows the positions of the amino acids, and at each position, we mutated the peptide to every potential residue, except cysteines, to prevent aggregation. So the white spots are those peptides that did not load, so could not be stabilized on the MHC. And the darker the color means the stronger the binding, the TCR for that particular sequence. And once again, here's some more quality control showing that the measurements with the standard SPR protocols are very consistent with this newer high throughput measurement. Okay, but the reason we're actually interested in obtaining all of these affinity values is so that we could measure killing against cells loaded with all of these different peptide mutants and correlate that with affinity. So now shown here in blue is not affinity, but the killing activity of different T cells against cancer cells loaded with each of these peptide mutants. So if you just focus on the wild type cells here, in the black boxes, I've represented the sequence of the wild type peptide. So cells engineered with this TCR can recognize a lot of different peptides, but they do have obviously what some people call a TCR footprint, and they have sequence preferences. So for example, at position 5, you can see there's a very strong preference for tryptophan, and mutating this residue to any other amino acid essentially abolishes T cell activation. Similarly, at other positions, this TCR can tolerate substitutions much better. So particularly at positions 1, shown here, and position 3, you can see there's a lot of blue. So essentially, you can mutate this position to any other amino acid, and the T cells will activate almost to the same extent. And then we compared this matrix to that of T cells when we knocked out CD8 in the middle, or where we introduced the co-receptor switch. And what I hope you can gather from this matrix is that there's a lot less blue in general than in wild-type cells. So they have a stronger preference for specific sequences. But interestingly, when you look at the black boxes, which is the cognate sequence, the activation against this target peptide is still very strong. And finally, we extracted the killing measurements from all of these matrices, and we could correlate them with the affinity measurements I showed before from our new method. And once again, you can see that CD8 knockout and co-receptor switch T cells can activate very strongly against the highest affinity peptides. But when we reduce the affinity, now it's only the wild type cells that can activate against those lowest affinity peptides. And what we did is, if we fit these lines and we extract an IC50, which is plotted here, is basically a metric of what affinity a particular T cell needs to activate on average. So wild-type cells activate about 50% of their maximal response with peptides of about 100 micromolar affinity. But when we knock out CD8, this gets reduced to about 10 micromolar affinity. And when we introduce CD4, this gets reduced again by about twofold. So this is just to say that obviously we are limited in terms of what number of peptides we can test for this therapeutic TCRs, but we hope that with this sort of unbiased approaches of modulating the co-receptors, we're truly just increasing the affinity activation threshold that the T cell needs to activate against a particular peptide. And this would reduce the cross-reactivities and hopefully improve the safety of the therapies. And that is all of the data, and I'm happy to answer any questions. I just wanted to thank the Duchenne group for all their help during my PhD, and specifically Misha and Anna, who performed all of the affinity measurements that then I used for my data. So yeah, I'm happy to answer any questions.
Thank you so much, Jose. It was really great for discussing with us this very clever way of basically making T cells smarter by guiding them to, react more selectively to their target peptides. Fascinating work as usual. Thank you so much. Just before we go through the questions, I have one short announcement, and that is from now on, because we live in the era of AI, we will have AI as one audience here. And at the end, I will read one question from AI. And you, Jose, as a speaker, I would like you to basically answer this in two stages. One, to basically comment on what type of question is. Is it completely out of context or rubbish or nonsense? Or perhaps it's a good question and actually, very relevant. And then for your answer, you can decide to answer, as a human is asking the question, or you can choose to basically say that, yeah, this is fascinating question, but I don't want to answer that. Okay, so it's completely up to you. And now I will start with our own questions. So please, if anyone have question, either raise your hand or please type your question in Q&A. I will start with a couple of questions myself, Jose. First of all, congratulations. This is really a fascinating, not only in the field of, therapeutics and medical application, but also enhancing the model development to understand T cell reacting to target antigens. So my very first question is that although the pool of target peptides that you basically considered in this study is very comprehensive, still, as you said, compared to potential target cross-reactives that sometimes one T cell can react to a million of targets, is very limited and very small. So how do you comment about that? I mean, you are not basically examining all the potential targets.
Yeah. No, that's a good point. I will also add, if a particular TCR binds with high affinity to what we consider an off-target peptide, there's nothing you can do about that. There's no affinity window there. There's no, a big assumption of all my work is that you will be working with a TCR that has been thoroughly characterized to be of very high affinity to a particular target and lower affinity to anything else. And that is usually possible for TCRs that are affinity matured, because it means that if the TCR is already supraphysiological affinity to the target, is unlikely that any other cross-reactivities will have the similar affinity. In terms of testing the pools of peptides and how to be comprehensive, I'll answer it in two ways. Firstly, ultra-peptide ligands obviously are not a metric of cross-reactivity, but of discrimination. So we can use them as a relationship in high versus low affinity and assuming that most off-target peptides are low affinity. Then, when we looked at these self-peptides, I'm happy to send you the paper, how these were generated, but essentially they use in silico prediction tools based on the TCR sequence to identify the ones that were most likely to activate to the TCR. And then this is the ones we tested experimentally, but we are sort of relying on their work to narrow the potential pool of peptides to test experimentally. And they did do that sort of with computational tools.
Excellent. The next question is from Ellen Burnett, and it's actually a very interesting question. It's asking really interesting research. I was wondering if there is concern that by increasing selectivity, there is a risk that cancerous cells will undergo a selective pressure to have a slightly modified target antigens that these T cells will no longer have high affinity to, and therefore be unable to activate against. Is the hope that with a large enough transfusion would be able to act quickly enough to avoid this?
That is a very, very good question. And actually, if we step back and think about other cancer therapies, like immune checkpoint blockade, you're actually trying to increase cross-reactivity in a way. So yeah, it's very, very specific to this sort of TCR transgenic T cell therapies where I think this could be useful. In terms of cancer resistance, the truth is I don't know. Of course, we show all of these peptide mutants. So yes, if the cancer were to mutate one of these, our engineered cells might not respond anymore. But then again, a lot of these mutations will not load very well on the MH as well. So I think it's not really something that is currently considered in these therapies as essential for effectiveness. When these therapies are developed, they are not trying to be cross-reactive to peptide mutants. They're not even, let's say, they're not targeted to NY-ESO. They're targeted to a specific 9-mer of NY-ESO. So we're already being very specific here. And what I would also say is for some targets like neoantigens, so for example, if you're targeting, I've talked mostly about cancer testis antigens, but if you're targeting something like KRAS mutations, driver mutations, you might actually only want to target a single point difference in the peptide. As in, you want to target a peptide that has a single point mutation, which drives tumor growth, but you don't want to target the wild type peptide, which is expressed by all healthy cells. So I think in this context, we do want to be very, very specific. But it is true that as for viruses with cancer, the cross-reactivity could help prevent sort of escape mutations. Maybe some of the findings from this work with CD5 we think could be useful in other therapeutic settings to actually increase T cell cross-reactivity to target cancer escape variants.
Thank you, Jose. Another question, in the examples of TCRs that you showed in this study, the TCR towards the original basically target, they had a very strong affinity to start with. But that is not the case for all T cells. I mean, some of them, they actually react in a very low affinity. So are there any chances of perhaps this strategy will either delay T cell response or basically reduce the magnitude of the response that we need?
Yeah, so this TCR, the C259, is actually affinity matured. So it has about a 1 micromolar affinity to NY-ESO. But the parental TCR that was isolated from PBMCs is actually about 10 micromolar. So it is, that TCR is called the 1G4 TCR, which we also use as a model system, but it was never used therapeutically. So that parental TCR, is about this range, right? 10 macromolar is already on the higher spectrum of physiological affinities, and that had to be improved even further for this therapeutic application. I understand there's a lot of TCRs that you might identify for specific cancer targets that are about 100 micromolar affinity, and this technology will not work for those because so 100 micromolar is 4A, 5Y. This technology will abolish activation against 100 micromolar affinity peptides. So it's only really targeted for high affinity interactions. Of course, if you have a low affinity TCR, you can always affinity mature it, but that itself is a whole potential cross-reactivity issue, because when you're affinity maturing the TCR, you're essentially creating a new binder with a new set of cross-reactivities itself.
Okay, I also had a hand raised by Stephanie, but I don't see her anymore. Perhaps, yeah. Can you demute yourself and ask your question? So let me check from here if I can do it.
Hi there. Great job.
Hello.
Great job, Jose. Awesome paper, as always.
Thank you.
I just had a question. Is this effect work within a certain affinity range for TCRs, or is it sort of a little bit of TCR specific where there's a window in which you can modulate that cross-reactivity? If it's too low, you're not seeing any effect because of, even the native TCR wouldn't be reactive, or if it's too high, you're not seeing an effect at all. Is that TCR specific in a way you might have to find that window per TCR, or is this, do you think, a universal rule?
Very good question. I would love to say it's a universal rule, but it's hard to compare on a same plot. I have a lot of data from other TCRs where the trend is the same, but you're right that I don't think I could just say for any TCR, anything above 1 micromolar is unaffected, and anything above 100 micromolar is abolished. It is kind of similar, but I think we would struggle to plot it on the same graph. And I think the reason as well is for different TCRs, we find that the relationship between sensitivity and affinity can be different.
Yeah.
As in two TCRs that bind a peptide with 1 micromolar will not necessarily have the same T cell sensitivity, which is something I don't completely understand the mechanisms of. But all the TCRs I've tested, the higher the affinity, the lower the effect, and the lower the affinity, the larger the effect. And roughly speaking, within 1 to 10 micromolar is very, very little effect or nothing. And I haven't seen anything where the peptide was over 100 micromolar and it wasn't abolished, if that helps answer your question.
Oh, cool. I mean, we're just trying to dig into it, right? Very, very cool.
Yeah.
Thank you, Jose, and thank you, Stephanie, for the question. The next question is from Brenda Road, and she's asking, I have rather a technical question. When transducing the TCRs on primary T cells, which kind of constant domain are you using to express the tag TCRs? Are you using a muralized TCR version to overcome competition with the endogenous TCRs?
So we use human constant domains. And actually, this was one of the reviewer revision experiments I've been doing, which I don't have it on the slides. We just use constant alpha, human constant alpha and human constant beta domains. We were asked whether mispairing with the endogenous TCR could affect our results. So I've done this month where actually I'll go back. with the same pipeline of producing the cells, I knocked out CD8 and the endogenous TRAC and TRBC locus at the same time, and then introduced our TCR with the transgenic TCR with the lentivirus. And what we found was a slight increase in the expression of the TCR, because it's not competing for CD3 components when the endogenous TCR. But when we perform these affinity discrimination assays, we obtain the same effect. So essentially, the activation wasn't changed. Sorry, the sensitivity was changed, but not the discrimination.
Thank you. If no more question from human colleagues, then I will ask the question from AI. So the question is that you have shown CD8 to CD4 switch enhances selectivity without compromising on target potency. Have you looked at how this switch affects activation kinetics or response magnitude in more dynamic systems like time course assays or in vivo? So first of all, what type of question is that?
This is the question we were asked by all reviewers. So it is
I thought so.
a very good question. And yeah, so I can answer the question. Yeah, we've only done peptide titrations at a single time point. So these are pulsed peptides on cell lines, and then we look at killing after 24 hours, but it doesn't give you information about the killing kinetics necessarily. So what we've done now, once again for the revisions, is to activate these cells against cell lines that endogenously present NY-ESO and measure the killing over time. And for different TCRs. So for example, I've done all the repeats now for MAGE. So we can show that knocking out CD8 for MAGE does not affect killing in this sort of more endogenous setting and more dynamic setting. And at different ET ratios as well. So going to low numbers of T cells, low numbers of T cells per cancer cell. Yeah.
Fantastic. So once again, because I don't see any more questions, once again, and thanks to Joseph for this very excellent presentation, great technique that you discussed with us, and it was a great discussion and fascinating questions. So thank you everyone involved for this discussion. And next month in July, we will have a speaker from BioNTech, so in industry-based strategy to generate some data and to answer an important question in mapping T cells to their target antigens. So I see you in 1 month time. Okay, thank you. Bye-bye.
Thank you. Bye.